This is Module 6, Eukaryotic Gene Regulation. In this video 6-1, Regulating Genes by RNA Degradation, I'll talk a little bit about the mechanisms involved in eukaryotic cells that target and bind to RNA transcripts, messenger RNA, and degrade them as a form of controlling gene expression. Eukaryotic cells and bacterial cells have a lot of things in common in terms of gene regulation, but they also differ in many, many different ways. And so eukaryotic cells and bacteria both regulate gene expression through the use of RNA binding proteins, for example. However, there are a lot of different ways in which those messenger RNAs are transcribed between eukaryotes and bacteria, eukaryotes and prokaryotes. Each structural gene in a eukaryotic cell has its own promoter and is transcribed separately. However, in bacteria and prokaryotes, each gene that encodes, so a structural gene is a gene that encodes a protein. Sometimes bacteria can encode multiple structural genes in a bacteria will all be under the control of a single promoter. And so what happens is one mRNA transcript represents 3 open reading frames. And so 3 proteins could be made from it, for example. However, in eukaryotes, every structural gene that makes a protein is controlled by its own promoter. In eukaryotes, DNA is packaged by wrapping DNA around histone proteins and condensing it into a chromosome, or chromatid. DNA, therefore, has to unwind from those histone proteins before transcription can occur. RNA polymerase, when it tries to bind to the promoter on DNA, won't be able to do that if that promoter is wrapped tightly around a histone protein. Prokaryotes don't have this problem because they don't package their DNA with histone proteins. So they simply must unsupercoil it or reduce the supercoiling, and then the RNA polymerase can access promoters in that way. And then transcription and translation in eukaryotes are separated in time and space, separated in space by the nuclear membrane. Remember that RNA is transcribed using DNA as a template in the nucleus, and proteins are translated using RNA as a template in the cytoplasm for a eukaryotic cell. So they must happen in different spaces, and therefore they happen during different times. Transcription has to happen first, and then translation will follow. In a bacteria, however, there is no nuclear membrane. They don't have a nucleus. I should stop saying bacteria, I mean prokaryotes don't have a nucleus. And therefore, transcription and translation happen at the same time in the same space. They both happen in the cytoplasm. And as an RNA molecule is being transcribed, ribosomes will already bind to the 5' end of the actively the RNA that's being actively made, and will already start making proteins before the messenger RNA is even done being made. And so they are coupled. They happen at the same time in this in the same space. And so because of these differences in how their genes are structured and how they're packaged, they have to regulate gene expression in different ways. I'm not going to talk too much about prokaryotic gene regulation. I actually talk about that in a different course called genetics. If you're a bio major, you'll get it later. Okay, so, excuse me. Some genes are regulated by RNA processing and degradation. And so if you want to express a gene, you have to access the DNA, use that DNA to create an RNA, and then use that RNA to create a protein. And so that whole process is referred to as gene expression. If you want to stop gene expression or slow it down, one way to do that is to degrade the messenger RNA. If you degrade the messenger RNA, then you won't be able to make a protein from that, but you still retain the information to make that protein in the DNA since we don't degrade the DNA. Okay, so one way in which RNA processing and degradation, so this slide is talking about RNA processing, and then I talk about how degradation of RNA controls gene expression. So first we'll talk about alternative splicing and how that how that regulates gene expression, sorry. We talked about alternative splicing already in the module 4, where we talked about RNA modification. But remember that alternative splicing can basically process a messenger RNA in one of two different ways, or maybe one of three different ways, and so on. And so first, we have this pre-messenger RNA that's been transcribed from a DNA template. And we have 1, 2, 3 exons in a single intron that separates exons B and C. One way to process this RNA is to splice out the intron and also splice out the B exon. And so A and C come together, and that gives you the large T antigen. Another way to do this is to just remove the intron. And so you get RNA A, B, and C. These three exons together will create the small T antigen. And so alternative splicing leads to the production of small T and large T. And so by regulating how this RNA is processed, we can either make the large T or the small T. This is done through the action of this SF2 protein. This is a regulator protein that enhances the use of the second splice site that you see here. And so in the presence of SF2, you will make more small T antigen. In the absence of SF2, you will make more large T antigen. And so we can regulate the production of these two proteins through the action of this RNA binding protein, SF2. Okay, so now let's talk about different ways to degrade RNA, since this is one of the major ways in which eukaryotes can regulate gene expression. Degradation of RNA can proceed in one of three different ways. Remove the 5' cap, shorten the poly A tail, or degrade the 5' untranslated region, the coding sequence, and the 3' untranslated region. So basically degrade the entire messenger RNA, chop it to bits. This slide right here is showing some of those different processes. And so this tends to happen in a stepwise process. So here we have a messenger RNA that encodes for a protein. This is the 5' cap as M7GPPP. This is the poly A tail at the end. And so the first thing that happens is deadenylation, or degradation of the poly A tail. Remember that both the 5' cap and the poly A tail participate in stabilizing the ribosome during the initiation stage of translation. And so if you remove the poly A tail first, and then degrade the 5' cap, the ribosome, when it tries to bind to the ribosomal binding site, won't be stable enough to migrate down the mRNA and find that start codon. So you're basically removing the two components of the messenger RNA that helps to stabilize the ribosome. If you remove them, the ribosome can't be stabilized, and therefore it won't be able to translate as much protein. And so you could stop there, where you just degrade the poly A tail. That will reduce expression. If you then decap it, that will reduce expression even further. Even though the mRNA is still intact, without the cap and without the poly A tail, the ribosome is not stable enough to actually make enough protein. Maybe some will be made, but not a lot. And then after you decap, and then the third level would be degrade the mRNA transcript altogether. Remember that the presence of the cap also protects the mRNA transcript from exonucleases that degrade in a 5' to 3' direction. Without the cap, exonucleases are now free to bind to this messenger RNA and chop it up to bits. And so through these three different actions, you can either reduce expression of a gene by removing the poly A tail, or removing the cap, or removing both. All three of those actions will reduce expression. And then degrading the coding sequence will obliterate expression. You won't get any protein because there's nothing to translate. Another way in which RNA degradation can control expression is through the use of RNA interference. RNA interference uses small interfering RNAs. This is what siRNA stands for, small interfering RNAs. So there is a DNA component here. So the DNA, it's not shown. So you have a DNA region of the DNA that is transcribed into this double-stranded RNA. And so the region of the DNA transcribes two complementary pieces of RNA, and then they find each other and will form this unstable double helix, double-stranded RNA. It's not stable at all. So this exists very shortly in the cell. If it exists long enough for Dicer to find it, Dicer will come and chop this double-stranded RNA into small interfering RNAs. And then it also chops them and makes them single-stranded. Those small interfering RNAs that are also single-stranded will be complementary to some portion of a messenger RNA target in the cell. And so that small RNA will combine with the protein complex RISC, and then RISC and small interfering RNAs will together scan messenger RNA targets in the cell looking for a complementary sequence. When it finds one, RISC, which is the protein involved here, will actually break the mRNA. And after this first break, that leaves it open to even further degradation by other exonucleases. And so the combination of RISC and the small interfering RNA will break up a messenger RNA target.